Good morning, good afternoon, and good evening, everyone. We are delighted to have participants joining us from across the globe. And we'll give it just another moment for everyone to settle in. I can see people still joining in. All right, so let's get started. On behalf of the ImmPort team, I welcome you all to today's webinar. My name is Sanchita Bhattacharya. I'm the Scientific Outreach Program Lead at ImmPort, and I'll be moderating the session today. A few housekeeping items before we go into the main presentation segment. The presentation time is approximately 45 minutes, followed by 10 minutes for Q&A. And please submit questions using the Q&A feature, and we will respond either directly into the Q&A window or address them live. This webinar is being recorded, and the recording will be made available afterwards. It is now my pleasure to introduce today's guest speaker, Dr. Anna Niarakis. Dr. Niarakis is a professor of bioinformatics and quantitative biology at the University of Toulouse. Her research focuses on computational systems biology and modeling complex human diseases, particularly autoimmune disorders like rheumatoid arthritis. She also leads the international working group building immune digital twins supported by the Research Data Alliance. Title of today's presentation is Digital Twins for Human Immune-Mediated Complex Diseases. With that, let's welcome Dr. Niarakis, and I'll turn it over to you, Anna.
Thank you so much, Santita, for the kind invitation and the introduction. It's a real pleasure to be here, and I hope that my presentation will spark interesting discussions and possible collaborations. As said, today I will try to touch a little bit on the concepts of digital twins for human immune-mediated complex diseases, and I will also present some modeling work that we have been doing in the lab the past 10 years. So the concept of digital twin, a very light definition, is that it is actually a virtual equivalent or a twin of a physical object. And these digital copies are increasingly popular because they can be used to drive important simulations that haven't been possible until now. The concept of digital twins is very intriguing and appealing for complex human pathologies because we want to organize and analyze biological data of different levels and different scales and organize them in computational models that can really scale. The results can help us with better therapies, understand response to treatment, treatment optimization, and also personalized medicine. The problem is that for the engineering to work, we need a manuscript. And the problem is that there is no blueprint for the human body and especially for the human immune system. So we know what we want to model, but we don't really know how the bits and bolts are put together in order for the system to work. Before continuing, I would like to say just a few words about these concepts of digital twins. So we have digital models, digital shadows, and digital twins. What makes a difference is actually the feedback that the model can receive from the physical object. We are in the era of digital models mostly, that we can make them represent or reproduce the behavior or the emergent behavior of a physical object, of a patient, for example, or a biological system, of a tumor, of a virtual joint. However, we have usually only one time point data, and the model and the data do not really talk to each other. Then we have digital shadows. A digital shadow is an evolving digital representation, is closer to the concept that we use in manufacturing for digital twins, and it can mirror the current state and behavior of a physical entity or system. However, the differences or the changes in the physical object cannot really can feed back to the model, but the model cannot have a direct effect on the physical object. What makes a digital twin is, by the stricter definition, a real-time connection between the physical entity and its digital counterpart. So a digital twin is a two-way information system that, in the best of cases, can handle a feed of data in real time. Digital twins simulate, monitor, and control physical objects or systems, facilitating analysis, optimization, and also predictive maintenance. So we have a live feedback loop, and this can foster insights about improving performance, efficiency, and reliability. An example of a real-time digital twin is the insulin pumps. They have evolved. I have no stakes in this company. It's just an example that I put in the slide because it's the latest generation of insulin pumps where you can have a wearable device that is connected to an application, and the model can measure in real time glucose on the blood, can calculate the amount of insulin that the patient needs to inject, and it injects it to the patient without need for the patient to do anything. The human immune response is omnipresent in most of the pathologies. So a focus on the human immune system is really justified, not only for autoimmune diseases, but also for aging, for trauma, and for infectious and emergent diseases. Using digital twins to improve how we care and how we respond to them can also give us more tools in our arsenal to tackle pandemics, threats, and be more prepared to face this type of threats that might be coming in the future. So this is a conceptual image that we published some years ago in the roadmap for the immune digital twins in 2022, where we show how different multiscale biological data can actually feed into computational models that can scale for data integration and analysis, and in silico simulations for diagnosis, modeling, prognosis, and also therapy optimization. However, the immune system is really complex, and even if we take abstract representations of it, it's not very easy to model it in detail or to understand how the different cells and the different molecules really interact and talk to each other. One good question that we always have when we start to build a model for a particular system or a particular disease is where to start. So I don't think there is a right or a wrong answer, but in the lab the past years, we've decided to have a middle out approach, start with pathways or cells, because this is where we can have most of the data, and this is where most of our tools work really well and can scale and can be efficient. And depending on the interest and also the availability of data, we can go either up towards population, so circuits, organs, organism, and population closer to a virtual patient, for example, or if the interest is really from, for example, from a pharmacological point of view, then we can go all the way to biomodules or proteins or even RNA and DNA when we are searching for targets and the way that they they behave with new medicines and new molecules. We use a lot network representations because they allow us to organize information in different levels, but they also create interfaces that can be biologically meaningful between molecular networks and cellular networks, but also between cellular and tissular organization. So networks feel very natural for biology, for organizing biological information, but also help us scale and better combine and integrate biological information at different scales. So what is the plan? The plan that we have been following, and you will see the results for some of the disease that we have been working with, is that we try to map the cells and the pathways involved in the pathogenesis or the mechanisms of interest. We try to construct causal networks. Causality is really important. So we don't really focus on co-expression or coexistence. We try really to reason by rich immunological, biological data and data sets to understand causality. We also do a lot of literature mining and reverse engineering, but also human curation to avoid false positives and a lot of noise in the structure and the topology, the backbone of the models that we build. Then we have tools and algorithms for an automatic inference of executable models, and we use them to predict, identify novel druggable targets, and also perform drug repurposing analysis. We use systems biology languages for representation, for the storage of information that can be machine readable, but also human readable. And we do it in a way that can be biologically meaningful. So we assemble the available fragmented knowledge in what we call disease encyclopedias or maps that can be manually curated or assisted and extensively annotated. And we use standardized systems biology graphical notation standards, stable identifiers to make these resources fair and interoperable. So here is just an example. You can have the ligand, receptor, complexation, the propagation of the signal, complex formation, gene expression, secreted proteins, and what we call disease phenotype. This is just an example to show you how the information can be organized, and we do this for thousands of interactions and molecules. So this is an example of the first rheumatoid arthritis graph encyclopedia that we created some years ago and we still maintain, where we have annotated exclusively all biomolecules and interactions using Miriam annotations, process description, and also PubMed IDs and stable identifiers using mostly data of human origin. These type of constructs use Google Map technology, so you can ask questions like, is there a pathway that can link interleukin-6 with an inflammatory phenotype? But you can also ask questions, you can search, and you can ask questions about the PubMed IDs, the type of data that were used in order to curate this interaction and to infer causality, and also to see if there are interacting drugs or interacting chemicals and microRNAs for the molecules of interest. These are interactive and freely accessible to the community. However, useful this type of representations, they are limited because they are static. So when multiple signals converge, it's very hard to understand what the decision would be for a cell. Let's hypothesize that this is a cell, for example, here. I don't know if you can see my cursor. And it's very difficult to understand what the cell will decide when multiple perturbations occur on the same time. So in order to have executable models, we develop tools that allow us to pass to a more compact representation and simulate dynamics by adding some mathematical description to these interactions. To make it a little bit simpler, here is an example where you have two activators and one inhibitor that can have a direct impact on the formation of this complex. Here you have, for example, different logical rules that are associated with it and the more compact network representation for the executable model. So now the inhibitor has a direct inhibitory action and the two activators have a direct positive activatory activity. So what changes is that we pass from biochemical reactions to activity flows to regulatory graphs. And we also infer logical rules that we write using mathematical symbols, but we can also translate in natural language, as you can see here. So activator 2 or activator 1 and the absence of inhibitor are sufficient conditions to have the complex formation or activation. So we do it for hundreds of interactions automatically, and the choice of the rules is based on semantics, topology, and experimental evidence. As you can see here, prediction is how the system will evolve in the next time step. So we set our experimental in silico experimental conditions, trying to imitate, to mimic experimental conditions in a cell culture or an in vivo model. And then we simulate what will happen when we do the corresponding perturbations. Depending on the rules that we will assign, we can have different results for the system. So when we are using AND gates, we are more strict. We ask for both A and B activators to be present in order to have C activated, while an OR gate leaves a little bit or flexibilities, less deterministic, so we can have either A or B as sufficient condition for the signal to be propagated. You can see different scenarios like one activator and one inhibitor, two inhibitors, and of course, this can be extended to multiple activators and multiple inhibitors depending on the system. What we do is that we perturb this system, we follow these rules, and we try to see if the system will stabilize. So we are using graph theory and mathematics and Boolean algebra to simulate all the state transitions of the system and see if there is a fixed point at the end where our system will be stabilized and cannot escape unless we perturb it again. So these stable decisions of the system can imitate phenotypes, stable phenotypes, hard commitments of the cell like apoptosis or cell proliferation, or oscillatory behaviors when we have attractors that are oscillating. So here is an example of three proteins or genes, A, B, and C. You see here that for A to be active, we need the presence of B. B is a positive regulator. For B, we need A and the absence of C because C is an inhibitor. And C needs A and the absence of B because B is an inhibitor for it. We can start with multiple initial conditions. A present, B absent, C present. Everything present and different combinations and start simulating. For these conditions, we can have a stable state where this will be the absence of everything at the end. So everything will be inactivated, starting with this perturbation. And for these, we will have an oscillatory cycle between these two conditions. And here it's just an auto-activating state. We do this for hundreds and thousands of reactions, and we also try to constrain the models using omics data or prior knowledge. So here is a computational framework that helps us link theoretical models like Boolean networks with discretized biological data. So we can use discretized expressions of gene expression datasets or classical molecular biology and cellular biology experiments, and we can use them to filter steady states and also to compare these conditions with the predictions of our models. How we discretize, this is just a very simple example. We can set a threshold for downregulation and upregulation and assign discrete values 0 and 1 to things that are not activated, downregulated, or upregulated. This is just one way. So to pass to more interesting things, applying them to real biological systems. The first model that we built was focusing on rheumatoid arthritis synovial fibroblast. So we were intrigued by the phenotype of this cell that was considered as a passive bystander for many years, but later publications and literature has shown that this cell has a phenotype, a more aggressive phenotype, closer to the cancer-associated fibroblasts. So they don't really want to die. They have an elevated proliferation rhythm, and they can also be controlling and sustaining inflammation if they are left untreated in particular pathologies like rheumatoid arthritis. So we wanted to build models to assess different phenotypes, and we wanted to do it in a modular way that if we wanted to study inflammation and apoptosis or cell proliferation and apoptosis, we could plug and play with the models. So we built large-scale models that were phenotype specific, but that they could be combined together based on the receptors and the part that is shared in the signaling pathways to see what was happening. We can simulate them separately, but we can also simulate all of them at once. So here are some probabilistic simulations. So now we are not in the binary, in the pure Boolean space, but we have added some stochastic noise, and we can use it to test input-output situations, like what is happening when I activate interleukin-6, what is happening to the inflammation phenotype, the same for growth factors and growth and survival, bone erosion phenotypes, matrix degradation, but also apoptosis. What is interesting in these systems is that they allow us to check not only input-output, but also intermediate states. For example, what is happening when I activate Fast ligand and apoptosis phenotype gets activated? Can I check what is happening in anti-apoptotic signals like AKT2? And this gives me a feeling of the credibility and robustness of the model, but also a way of monitoring the system and make correspondence with real-time metrics of the system and experimental data. So we used it in order to model the effect of mono and combined therapy for rheumatoid arthritis, especially for guiding and designing experiments for fibroblasts. And some of these predictions were validated in vitro, and some others are ongoing. Then we wanted to add metabolism. So metabolic switches and metabolism and inflammation are also emerging fields. So we wanted to see if these cells, rheumatoid arthritis synovial fibroblasts, undergo under hypoxic conditions of the arthritic joint metabolic reprogramming as their cousins in the hypoxic environment of the tumor microenvironment. So what we did is that we devised a hybrid, we proposed a hybrid methodology in order to couple signaling and genome scale metabolic models. So what we did is that we took our logical model, our Boolean network that was addressing signaling and gene regulation, and we found ways to couple it with the metabolic model. So what we did in order to see if it works is that we simulated our model. We saw where the model stabilized, and we took these stability points, these fixed points, as initial constraints to launch flex balance analysis. When we did it without any constraint, as you can see, the total production of ATP for this cell is through TCA and oxidative phosphorylases, which is a hallmark for a healthy cell. And when we applied the constraints that came from simulating a rheumatoid arthritis synovial fibroblast model, without touching anything else, we saw that now the ATP, the production of ATP, was not through TCA, but was through glycolysis. So this was very close to an inverse Warburg effect. And then we performed knockout simulations, exhaustive simulations in order to identify the switch, the culprit for changing this behavior. And as you can see, through these simulations, we could identify HIF1 as the target that could change, that could reprogram fibroblasts in order to change their metabolic program. And completely independently, around the same time, experimentally, this was shown for healthy metabolic profiles in rheumatoid arthritis fibroblasts by targeting HIF1. We also use these approaches to target macrophages in rheumatoid arthritis. We used two extreme phenotypes, M1 and M2, largely because we didn't have enough data to study the polarization. So we took the two extremes, and what we wanted to do was to see if we could press the system to have more anti-inflammatory phenotype, more M2 and less M1 phenotypes in the articular joint. So we wanted to have selective M1 macrophage depletion and selective M2 macrophage promotion in the RA synovium. So for us, this was very close to conceptually to identifying switches that could change the transcriptional programs of these cells. So we did exhaustive simulations, and we found, for example, for M1, between proliferation and apoptotic phenotypes, NEFKB inhibition could help us change the profiles for M1. We also did find GSK3B as a therapeutic target in order to change the profile between proliferation and apoptosis, but for the inverse outcome. And we also did combinations like ERK1 and NOTCH1 that have been tested in cancer settings, but never in autoimmune disease or rheumatoid arthritis. We also tried to investigate a mode of action, especially for baricitinib, that showed an effect on macrophages phenotype and able to change the program, the proliferative program and apoptotic program on the M1 populations. Going one step further and collaborating with industry, we decided to build a multicellular model for rheumatoid arthritis. So we had M1 and M2 macrophages. We already had a fibroblast model, and we wanted to see if we could develop a T cell, a more generic T cell model, and put them all together. So what we did is that we tried to see how we could put these cells talking to each other and simulate them on the same time, having as outcomes global phenotypes for the articular joint. We had many challenges, especially for the scale and the number of nodes and interactions for each intracellular model. And we also had difficulties in identifying proper data sets in the public space that could allow us to decide this vocabulary, this crosstalk between the cells, resident cells and immune cells. I won't go into the details. All of this work is already published. All the code, all the models, all the resources are open access. And if you're interested in implementing some of this work, don't hesitate to email me for the sake of keeping it lighter from a technical point of view. I will not go into these more technical details. So just to give you a feeling, the size of the models are not negligible. So for fibroblasts, we are around 1,000 species. For M2 macrophages, 500 species. For M1, 650. And the smaller model on the smaller side is the TH1. We used co-culture data from co-cultures and also statistical methods in order to deconvolute signals and understand better how these cells might talk to each other in the arthritic joint. So we developed the model that we used to simulate. The model was huge. It's one of the biggest models for a biological system using the Boolean formalism. Is 1,204 nodes, and it is fully accessible and simulatable. So we used it to investigate new therapeutic options. Disclaimer, some of these I cannot really disclose, but I can show you results from the calibration with data from the public domain. And this is why it's so important to have rich data sets in the public domain, because they help us really advance and validate predictive models and hypothesis testing. So published experimental evidence supported many of the targets that the model predicted. Here, for example, it's the inhibition of TBX21 for down-regulating hyperactive RATH1 cells. We also reproduced CAV inhibition for hyperactive RA fibroblasts and mere 221 inhibition reducing angiogenesis with subsequent activation of THBS1. We also use the model to investigate new therapeutic options and combinations. Here, for example, you see the in silico knockout of NF-κ beta and STAT3. That has a direct impact on the osteoclastogenesis. Here are a combination of targets that could have a direct impact on cell-specific phenotypes, but also global phenotypes, and a list of targets and repurposed drugs, drugs that have been approved and passed clinical trials for different disease, but never used for rheumatoid arthritis. So some of these targets and some of these combinations are tested as we speak in vitro and human-based assays. So as I said, all this work is already published, fully accessible, and we would be happy if people would be interested in using some of it. So the past couple of years, we have been really interested in developing digital twin technology, meaning scale even more and not have cellular or multicellular systems, but pass to tissulars or even 3D systems like a 3D articular joint. So very recently, we received a very competitive funding to build a digital twin for personalized treatment of rheumatoid arthritis in collaboration with the rheumatology department at the Hospital of Kremlin-Bicêtre in Paris and an experimental partner using spatial transcriptomics and technology at Genoscope in Evry. So the idea here is that we take these very big models and we use them as brains, as control and decision systems for the cells. So now we will not be modeling molecules, we will be modeling cellular crosstalk, and these individual programs that will dictate the behavior of the cells will be simulated separately. So this will allow us to have intracellular networks, keep molecular interactions, keep qualitative and biochemical reactions and metabolism if needed, keep signaling and gene regulation, because this is where the drugs act. We need the targets, and then have cellular crosstalk, spatiotemporal aspects, and a capacity to introduce biophysical features, because we also want to start approaching the clinic and what a clinician or an experimentalist can actually use or see. So this is a 2D architecture of an articular joint where this is, for example, the pannus formation. This is the bone with chondrocytes and collagen, and this is the cartilage with chondrocytes, and here we have fibroblasts and macrophages, and here we have the synovial fluid. So we can simulate, we can go all the way up to 50,000 agents using a laptop and 500,000 agents using a cluster. And with that, we can simulate cell populations and cell crosstalk, keeping things neat and kind of separate from the intracellular networks that we simulate separately. We also we don't work only in rheumatoid arthritis. For the sake of time, I would like also to present some results in different pathologies. We also work with autoimmune syndrome of Sjögren, which is now Sjögren disease from last year. So the idea was to map inter and intracellular pathways, map epithelial cells and their relationship to immune cells and especially B cells, extract data from literature, use language parsers and probably AI assistants, but also human curation, and also programmatically assess databases like Reactome, KEGG, and Wikipathways to assemble pathways of interest programmatically and more quickly. So we collaborate with the Necessity Consortium. We have access to cohort and clinical profiles, but we also have a collection of very heterogeneous data in blood, in salivary biopsies, in cultivated epithelial cells, in things that are in vitro, in vivo, ex vivo, and also transcriptomics in different layers, single cell or bulk with different technologies. So integrating data is also very challenging. However, we are now in the process of publishing a big part of this work that has been going on for the last 4 years. These are some unpublished results for experimental evidence, especially for interferon type 1 and 2 simulations, and what is happening when we're adding different drugs. And also unpublished results for targets, drugs, and drug combinations that we are now revising to see how many of them are actually clinically plausible and how many of them can be tested in cell-based assays or organoids at first before considering moving forward. We have also been working the past couple of years in atopic dermatitis. We started with bibliometric analysis and extensive literature analysis. We have built conceptual models between 12 different cells, 36 entities, and 58 interactions, causal interactions that have been validated experimentally between sensory neurons and immune cells, cells and molecules, and cellular crosstalk in different levels. We have put together this vocabulary, this resource of communication between different cells of the immune system and resident cells in the settings of atopic dermatitis. And we have, this is a conceptual model for the epidermis, the dermis, and the blood vessel, and how different cells communicate with each other and how different perturbations can have effects in pruritus or itch scratch, in hyperalgesia, in neural hyperexcitability. So we are trying to make the link between molecular, cellular, and phenotypic full body manifestations of the patients. We are also analyzing single cell transcriptomic analysis datasets in human and in mice. And we are also using transcriptomic, spatial transcriptomic. For this modeling aspect, we are trying to have a 3D reconstruction of the skin with portfolios of different cells that talk to each other. For example, here we have a population of a sensory neuron, keratinocytes, and mastocytes. This is for presentation purposes, this is not fully calibrated, just to show you what can be done. And these populations can behave can be contextualized according to expression data, and they can actually show expression for different biomarkers, and you can have the results as temporal dynamics. Here we have filaggrin expression for keratinocytes in healthy conditions, and here keratinocytes in atopic dermatitis with filaggrin, interleukin 25, 33, and TSLP. And you can have quantified results, but also temporal dynamics, where you can see the pulses. In a healthy system, the pulses are really spaced between them, where when we have excitement of the system, you can have this oscillations and this temporal dynamics for histamine, tryptase, and different interleukins. We are also analyzing spatial transcriptomics, as I said, in human, but also in mice. And what we want to do is that we want to approach the architecture of the tissue with our 2D or 3D model reconstructions. So for the moment, we are analyzing also mice studies. We have the first batch of 4 mice, a model of contact dermatitis, and we are also expecting a batch of 4 mice of the oxazolone model, and we would like to move forward with that. In order to leave some time for the discussion, I would like to leave some points about the added value of the in silico part. Many people ask, what is the added value of having a computational model, an in silico part along the in vitro, the in vivo? So the dynamic aspect is really important. It lets us do predictions and hypothesis testing. We can prioritize things for you. We can also help you skip the lists or the Excel files. Having a list of upregulated or downregulated genes or biomarkers don't say much if they are not put into their context. So you probably know it better than me. In a living system, there is nothing that acts in isolation. So having networks, causal networks, mechanisms is really important because it really deepens our understanding of the system. We can accelerate the research by prioritizing targets and combinations. We offer complementary approaches, and this is very important to stress that we like to work with experimental partners and also clinicians. The computational and in silico part, computational biology is not another biology. It's the same biology. So we really need to be together in this aspect. The methods are usually disease agnostic, and we can use them or even contextualize and personalize the models depending on the data availability. We also work in formal aspects in order to scale and handle the computational problem. These scales are not very easy. Even for binary systems, when you have more than 1,000, you have 2 in the power of 1,000 possible states. This is not possible to analyze at this point. So we need formal methods that help us downsize the complexity to make it manageable without losing track of the richness of biological datasets. Limitations, data are available, but data are wide and not deep. In many cases, we have many different data sets that are for one point, for one time point, or not for all patients, or different platforms. So we really need to address statistical power, noise, and data cleaning and integration. And this is why initiatives and portals like ImmPort are so important. Depending on the approach, quantitative or qualitative, we adapt, and we also take care of interoperability and fairness to be sure that we create models that can be reusable and accessible, and also try to leverage the computational demands so that these models are accessible to the right audience, experimentalists, clinicians that are interested in implementing this type of in silico branches to their research projects. And for digital twins, how far are we? We have some successful examples in diabetes and in cardiovascular diseases. And I'm really hopeful and optimistic that in the next couple of years, we will have more successful examples, especially in the space of autoimmunity. We also have artificial intelligence that is developing fast, and causal AI will play a major role. I'm not talking for LLMs, but I'm also talking about reason, AI that can reason. Scale up can be tricky, and we also need to address ethical aspects, democratization of this technology, but also keeping a human in the loop so that these models are accessible, but not too accessible, because without validation, there is also the cost of liability and the possibility of errors. And with that, I would like to acknowledge everyone who has worked with me, collaborated with me over the years, clinicians, industry, students, of course, experimentalists, theoreticians, mathematicians, informaticians, everyone, and also the students who have done most of the hard work and the results that I have presented today. And with that, I would like to finish with saying that we have an international group of building immune digital twins supported by Research Data Alliance. We have our portal that is being developed as we speak to make these models more accessible, and we would be delighted to have you joining the community. Thank you for your attention, and I would be happy to receive questions. So.
Wow. Thank you, Dr. Niarchis, for this excellent and thought-provoking presentation. I could
Thank
see
you.
that. Let's open the floor for questions. We already have several questions coming in. So let's begin, and I'm going to read in the order we received it. The first one is, applaud, nice work. How was stochasticity added in the dynamic models? And is it the network same for different cell types?
So the networks are not the same for different cell types. The networks are different in topology and they are different in specificity. So they are fully contextualized. We have not used a blueprint of a cell and tried to kind of accommodate it to meet different cell type criteria. We have developed them from scratch using data that are cell and disease specific to the measure of possible. So regarding stochasticity, we used stochastic simulators. So for some of the results that I showed, we used MABOS, a stochastic simulator using Gillespie algorithm that was developed from the Institute Curie. But we also have in-house tools that we are building to add more stochasticity or change a little bit the parameters. And also a simple stochastic simulation could also be achieved with some in silico platforms like Cell Collective that are more accessible they are web-based and more accessible for less computationally savvy users.
Great. So maybe I'll move to the next question. Boolean mechanism capture all or none models for individual variables. How do you
Yes.
model gradient and or longitudinal variables?
Yes. So it comes with limits. Huh? It's not perfect. We use hard binaries in some cases, but we also use stochasticity, so probabilistic frameworks for some of them. We also use different updating schemes, synchronous or asynchronous, and we introduce delays and weights. So we try to be as less strict as possible to approach a biological system. And depending on the interest, for example, often we use large-scale biological networks that are fully binary because we can scale them. And once we identify targets and mechanisms of interest that are very specific, like the switches that I showed you previously, then if we have more time series or quantitative data, we also do OD-like models that are more detailed and quantitative and allows us to be more precise. But these type of models do not scale well, so we cannot use, for example, 500 ODs. It cannot be possible. So we leverage depending on data availability and interest.
Thank you. And we go to the next. Applaud. Thanks for this exceptional presentation and wonderful research. There's a
Thank you.
I think it's more a question or clarifying that what if analysis simulation, have you considered incorporation of AI into this simulation? It seems that AI
So,
models can take much of the burdens in simulation, and it cannot take the full
so
responsibility of whole simulation.
Yes. So we are carefully examining what type of AI we are going to introduce into this framework. So for us, keeping the mechanism AI free is very important because it allows us to have a very elevated confidence level regarding the mechanism. But it takes time and it scales poorly. So we want to see, so we are using AI assisted curation, for example, which performs really well. It has less hallucinations as it had when it started, but it still takes a lot of time to check. For us, it's very important to have a cross validation. So even if we do use AI and we accelerate this part, we will also do a validation with an external data set, whether this would be an omic data set or an experimental data set of low throughput, because we want to be sure about the causality. So this problem is not fully resolved yet in AI reasoning, but I think we're getting there faster than we thought. So yes, this is definitely something we want to do and take the burden not only out of the simulation, but also of the model building. Because for us, it's not the simulation that takes a lot of time. It's the actual model building that can be tedious.
Hope that answers the question. Thank you. And let's go to the next. Moving from molecular or cellular to clinical practice and learning health system perspective, your insight generation happens in the digital twin end. However, data are streamed through the shadow component. Have you considered utilizing edge computing to generate insights closer to devices and sensors to eliminate the step of storing raw data for future processing for digital twin projects? And would, doing so would shorten the time lapse between the signal detection and response or recommendation. How
Yes.
feasible do you think such edge compute approach is?
So this is an excellent question, and thank you for bringing this up. So for me, this comes from there are two, I would say, groups in digital twins. The ones that come from molecular, cellular, even tisular, and the others that come from clinics, patient, device. And we are trying to see how to reconcile both of them because we need both of them. The problem with devices is that they don't give you any information about the mechanism or treatment optimization. And on the other part, molecular targets do not tell you much about the full body manifestation or the clinical measures that the clinician will take. So it depends on the disease and it depends on the feasibility. For brain diseases, for example, we cannot have a sensor monitoring the brain. In rheumatoid arthritis, we don't have good biomarkers circulating in the blood. And it's very hard to also have biopsies. For example, in certain countries, especially in France, biopsy is considered as very invasive, and it's not part of the clinical practice of let's say everyday clinical practice. You need to have a very good reason in order to do it. And then we are also lacking ethical and regulatory aspects that would regulate the access to data and also making sure that these data are secured in the streams and that the patient is well informed. Not many people would like to wear something and give away their data 24/7, or maybe they would, but we are not yet there. And I think what you are proposing is absolutely correct. For the diseases that I'm working with and from the scales that I'm approaching them, this will come a little bit later for me. But for cardiovascular diseases, for liver diseases, and for diabetes, these discussions and applications and the digital twin, let's say, community are already using this type of approaches to eliminate lapses and have a more rapid signal between the device and the digital shadow.
Excellent. I do see a
I hope I
comment
answered.
on digital biopsies. The comment is that digital biopsies are not invasive and are underutilized.
Yes, this is true. And also X-rays or anything that could help us have, for example, non-invasive things that happen in the clinic and we don't have access to it. So, for example, it's very hard to have access to this type of information. And for osteoarthritis, for example, a huge data set was released recently, 1 year ago, to help build machine learning and AI models. And we're hoping that the same would happen also for other types of diseases, for rheumatoid arthritis, for example. So I totally agree. And imaging is also a source of information where AI has done miracles.
Thank you. And we still have time, so let's take this next question. Great talk. As a statistics researcher and a newcomer to this field, what is currently the biggest challenge in modeling? Fusing multiscale data, computational efficiency, quantifying uncertainty, prediction accuracy, or accounting for complex interactions between cells.
I would say all of them, but if I had to pick one, I would say uncertainty quantification. Because if we really want to move from models that we build for research and models that we test in vitro and cell cultures, and we really need to think of how to reach the from bench to bedside, I think that we truly need to address uncertainty and credibility. How to make models robust enough, how to make models causal enough, how to build models that are not useful only for one question, move away from the dogma of computational biology, where we were building one model for one question and this model was never used again. I am a fervent advocate of building reusable models that we can share and models that can be contextualized. And this is why I support standards and fairness. And if I had to pick one, that would be credibility and uncertainty quantification. The technical and more formal aspects, we will get there sooner or later. But these things need to be addressed before arriving to the final destination that is the patient.
Thank you. I think there's a follow-up question on that, which was also something I was thinking of asking you. Are there any suggestions for publicly available data that could help developing these models to address these challenges? And my interest would also be about like, like have you done any kind of this kind of a data analysis directly from the public repositories, downloading the data and running the models?
For 10 years, my research was fully related and dependent on data on the public space. It has been the last 4 or 5 years where the mindset changed and more data sets were available. And we also had more access to clinical cohorts, patients' data through interdisciplinary projects because clinicians and experimentalists were more open. For many years, the mindset was that bioinformaticians or computational biologists would be the last person, to talk to, where you would just dump the data and say, make something out of it. While now, computational biologists co-design the projects and the experiments. When we started building the first model for fibroblast, there was only two data sets with 15 patients. So someone asked me, why do you build a mechanistic model and not a machine learning one? And I responded then, how can I build a machine learning model with 30 patients? So nowadays we have hundreds, and these are fully accessible for models to be calibrated, trained, and tested. So yeah, we definitely rely on data in public repositories. And just a last comment, when we work with industry a lot, we make sure that the technology is also accessible and open to the scientific community. And in order to do that, we need to calibrate them with publicly available data. Otherwise, the model calibrated with proprietary data will never see the light of a publication. So having good data sets are really important for the openness and advancement of the scientific community.
Thank you. I think we are going a little bit over time. Thank you very much. It was a fantastic talk. And
Thank you so much.
I also thank everyone for joining us today, for thoughtful questions, participation. And if we were not able to address any of your questions during the live session, or please feel free to follow up. And I think Anna, you will be happy to get emails and any questions people might have. And this just
I see one question for the slides. So I
yeah.
will clean them a little bit for the unpublished things that I'm not really authorized to share right now, and I will give you a full deck minus 5 or 6 slides that you can share with the participants.
Thank you again and have a wonderful
Thank
rest
you.
of the day. Thank you. Bye bye.
Thank you. Thank you so much.